Good afternoon, everyone. I'm very happy to be here today to talk about the project we have been working on for the quantitative linkage between TTP and time to culture negative status to optimize drug development decisions. So there is currently a need to develop quantitative tools to more accurately evaluate efficacy in phase 2 clinical trials for combination regimen for tuberculosis, and also for more reliably predict clinically relevant endpoints for phase 3 studies. So the linkage between early biomarker measurement in phase 2 studies and the long-term clinical outcome in phase 3 studies may help increase efficiency for drug development in TB regimens, for TB regimens. And if this linkage is successful, this novel approach may reduce the time required to develop regimens from decades to years. With the culture-based evaluation of tuberculosis, a parameter has emerged as an alternative to solid media cultures, and it's called time to detection, and also known as time to positivity, TTP. So TTP represents the time to detectable growth of Mycobacterium tuberculosis in culture. And the potential use of TTP as an early indicator of treatment efficacy comes from an early work performed by Epstein, who showed that TTP of Mycobacterium in sputum culture correlates with the response to anti-tuberculosis therapy. So the goal of this project was to evaluate the relationship of TTP trajectory parameters with clinically relevant endpoints in phase 3 trials like durable cure and relapse, based on data from 11 phase 2 studies and the REMOX trial, and also to create a biomarker disease model by linking the above models. So the biomarker disease model would be a parametric survival model driven by the TTP specific parameters. So if we look at the, is there a pointer? No. So if we look at the graph on the left, the TTP specific parameters, that would be like if we take one of those parameters, and I will go in more details in the next slides, like the gamma, which is the rate of growth. So the line in blue, which is high gamma, faster rate of growth, would translate in the survival model in a faster time to conversion to negative growth. So this is what we will be showing, like how we would be linking the TTP parameters to the disease model. So the data we have used is the REMOX data. This is a phase 3 trial design. It had 3 arms, the HRZE, the control arm. It had another arm, which is the HRZM, where the ethambutol was replaced by moxifloxacin, and a third arm where isoniazid was replaced by moxifloxacin. The biomarker we would be using from this trial is the TTP, time to positivity, and the endpoint would be time to culture negative status, which is defined as 2 negative culture results at different visits without an intervening positive result. And the time to negative culture would be the time of the first negative culture. So the TTP data was described by a Gompertz model. Just for clarification, this Gompertz model was developed based on the 11 phase 2 data, and then it was applied to the REMOX data. What's important in this model, it has 3 parameters, like I'm showing in red, alpha, beta, and gamma. The alpha would be the maximum value that can be reached. In our case, it's 42 days because the cultures were observed for 42 days to say that if there was a growth or no growth. After that, they were they wouldn't take the results. It would just be considered no growth. And then the beta, which is the TTP at baseline, so the baseline value of TTP, and gamma, which is a constant related to the proliferative ability of Mycobacterium tuberculosis in culture and rate of, it's kind of rate of growth. And the reason this parameter is important is that we can translate it to a half-life. Like talk about half-life to reach the maximum effect. And this is example how changes in the parameters of the Converse model could affect the shape of the TTP profile. So if we look at the graph on the left, so this is a TTP profile for different values of baseline. The value in green would be a low baseline, and the value in blue is high baseline, and the red is the mean value. And so the beta changes in the beta values, like from low to high, would make the curve move from the right to the left without any changes in the steepness. However, if we have changes in the gamma from low values like green to the blue, so this would affect the steepness of the curve, so the rate to which we reach the maximum value. So higher values of gamma means faster rate to reach the maximum value. And based on these observations, it was hypothesized that these parameters describing the TTP profile could be predictive of clinical endpoint in phase 3 trials. So this model, developed from the phase 2 data, was applied to the REMOX trial, and we were able to derive the TTP growth, so the gamma values, the last line, and also derive the half-life for each of the treatment arms that were used in the REMOX trial. So for the isoniazid arm, the half-life was 14.4 days. For ethambutol, it was 13.3 days, and 15.8 days for the control group. And then we used this data to do a survival analysis, and the values I have just shown could be translated in this graph to only slightly better performance or faster time to culture negative. So it's the red and the blue lines compared to the green line, which is the control group. And then, so this is based on the data for 12 months. But instead of showing this analysis by treatment arm, we divided the data by according to the values of the gamma, the rate of growth. So we divided the data. We had 3 groups. So people who had a low rate of growth, people who have like mean or median, or the group in the middle for rate of growth, and people who have high gamma, faster rate of growth. So the faster rate of growth in the figure would be in green, and the lower rate of growth would be in red. So we did survival analysis based on this stratification, and we clearly see from this survival analysis that patients who have a higher rate of a high gamma, so higher rate of growth, would have a faster or a shorter time to conversion to culture negative status. So it's the line in green. And if we derive a half-life to reach to this time to conversion to negative, it would be 22 days for subjects in the high gamma group, and 44 days in subjects who have a low gamma. So everyone starts at the 100% probability of having a positive culture, and we aim to have these subjects have a negative culture results. And this analysis was based on all data, so data collected from time 0 to 12 months. But then we were interested to see the effect of the duration or time of collection of TTP data on the prediction of this analysis and prediction of half-life. So we only selected TTP data from 0 to 8 weeks and redid the same analysis. So we took only data from 0 to 8 weeks, derived again our gamma, stratified patients according to their gamma group, and did the analysis, and we get the same results. So again, 22 days is the half-life for patients who have high gamma, and 44 days for patients who have a low gamma, so a low rate of growth. So the TTP data from 0 to 8 weeks was as predictive as the data from 0 to 12 months. Same analysis was repeated, but again was having a different subset, so only data from 0 to 4 weeks. So in this analysis, again, we got the same results for the high rate of growth group, medium rate of growth group, but for the low gamma, low rate, we had a slight change from 44 days to 42 days. But again, this was like predictive of the performance we had with the 0 to 12 month data, and was telling us the same, that the higher the gamma, the shorter the time to conversion to negative results. Again, we only took the data from 0 to 2 weeks, but this time we saw a loss of resolution when only considering time from 0 to 2 weeks. So the values for the high, so if you see the curves, like if I go back, we have a clear difference between the three groups, but in this one, the curves are very close to each other. It's harder to distinguish. And the half-life predicted, like for the high group, high gamma, changed from 22 to 28, for the medium group from 36 to 35 days, and for the low gamma, it changed from 44 to 36. So we were not able to get the same values at the data from 0 to 12 months. So to conclude, so the linkage between early biomarker measurements in phase 2 and long-term clinical outcomes in phase 3 may help increase efficiency of drug development. And the rate of TTP growth, as per the Compert's model, is a potential marker to use as a prognostic for prognostic biomarker for response. And also the early part, so 0 to 4 weeks of this profile, is very informative for clinical endpoints. The potential application and value of this work are to determine early changes in TTP in phase 2, like in dose ranging studies, and effect on long-term clinical outcome in phase 3 to guide decision, and also to inform gate decisions when considering advancing from phase 2 to phase 3 data. Thank you for your time, and I would like to thank also my colleagues, Jeff Marier, Dave Herman, and Samir Moksasi from Certara, and Klaus Romero from Critical Path Institute. Thank you.